Thank you very much, everybody, to be here. So it's a pleasure for us to introduce our talk, and the title is How to Overcome the Challenge in Highly Potent API in Chemical Development. Hello. So first, one introduction. I am Jean-Guy Boiteau, Head of Chemical Development at Nuvisan.
And I'm Thibaud Gerfaud, the Head of Process Research here also at Nuvisan in Sofia in France.
Some rules first to start and to have a good presentation. So please be aware that you have to switch off your microphone by your computer. You will have to use the question and answer chat box, and all the recording will be available just after the presentation. We are going to start with a poll. I think there is a question that will be raised, so you have to answer on this slide. Did you collaborate with Nuvisan before? You got several choices. Give me the go. You have to wait 1 minute. Okay. So we can go directly to the. Do you have any results right now? So, no, I never work with Nuvisan, so yes for chemistry at 14%. So it's a nice introduction. As many of you do not know Nuvisan, it will be a nice thing, in fact, to introduce our site and what are our capacities. So Nuvisan is in fact the former Galderma site in the south of France. And in 2018, 82 people joined Nuvisan, and especially in the teams of chemistry, formulation, manufacturing of clinical batches, analytical service, bioanalysis, and also regulatory and quality. The agenda for today's talk is some introduction. We will explain, in fact, our legacy and especially why we are talking about highly potent APIs. Then we will explore the classification of these APIs, the OEB banding, and some process development challenge. And at the end, we will have the Q&A session. First of all, as an introduction, I would like to introduce the CMC capacities at Nuvisan. In fact, it can be divided into two different big parts, the discovery and the development. And we can work since the really beginning from target identification to some medicinal chemistry, chemical development, preclinical profiling, and also radioactive synthesis. So this is all we can do at Nuvisan for the chemistry activities. And we will focus especially on this talk on the chemical development. And what we want to do at Nuvisan, south of France, is to bridge the gap in between the CRO and the CMO. And we can manufacture, in fact, the API from the road scouting to phase 2 batches. We can develop the synthesis from milligram to kilogram scale. We are GMP compliant. We can manufacture tox and GMP batches up to 5 to 10 kilograms. It will depend on the solubility of the material. Usually, we worked with 3 different work packages. The process research, where we are working on gram scale, usually at the bench. Then we can move to the process development. Usually it's on 100 gram scale. It's a rough idea. And after this step, when the synthesis is complete, robust, and we believe that it will be transferred to the kilo lab, we can manufacture the GMP batches on kilogram scale. So first of all, some definition. When we talk about highly potent APIs, which means molecules that have a high potency. In fact, it's a high potency that produces an effect at very low dose. But if you say high potency, you have also to say high toxicity, because the toxicity is also low. And we can see some different drugs that we can have on the market, the Doliprane, Singulair, or Lilu. It's this compound, in fact, you can have 1 gram inside, 10 milligrams or 20 micrograms. So you can see that the potency of each molecule is very, very different. And that means that the toxicity also is very, very different. I would like to introduce our retinoid legacy. As I said at the beginning, we were the Galderma laboratory and we work a lot on retinoids. And the issue with retinoids, in fact, is that the target of retinoids is the skin and also the keratinocyte. That means that if you are exposed to a single particle, in fact, of this kind of drug, depending on the potency, 24 hours to 48 hours after exposure, you will have a skin erythema, a sunburn, a peeling, and your skin will be completely removed, and you will have a complete new skin. So it's maybe why we look so young, but this is another story.
Okay, so now let's focus on the classification of APIs and HPAPIs. So obviously, all the classification is done thanks to the tox studies. So the tox studies are performed, reviewed by toxicologists, and that helps them to calculate what we call PDE, so the permitted daily exposure, which is the dose that is unlikely to cause an adverse effect if an individual is exposed at or below this dose every day for a lifetime. So that helps to protect the patients, to define the dose that will be used in the clinics. And for us, as chemists producing the APIs, that help us to define the cross-contamination level and the cleaning methods. And from these PDE, we can also derive OEL, which are the occupational exposure limit, which is the concentration of an API in air that an employee can be exposed during an 8-hour workday without presenting health hazard. So it's a value in microgram per meter cube, and that helps us to protect the people preparing these ingredients. And we are lucky enough at Nuvisan to have our own toxicologist, Nathalie. So she's able to review all the data from the literature or from our clients and calculate PDE, OEL, and classify HPAPIs from regular APIs. The problem for us at early stage chemical development is that we are the people preparing the batches that will be used in the tox studies. So most of the time, and for all new chemical entities, as a CRO, our clients usually do not have any data on their molecule for this, the production of these early development batches. And we need anyway to ensure an adequate level of protection between the workers, and for the patients. And we don't know the exact nature of the compounds that we are producing. And there is another issue here, which are that the OELs and the tox studies are all conducted obviously on the final API, but that does not give us any clue regarding the synthetic intermediates, so the molecules that are isolated along the synthesis of the API. So we need really to find the right level of protection. We do not want, obviously, to be like this, fully unprotected and manipulating dangerous substances. And in the meantime, we do not want to be taking over precautions and running NMRs, wearing a vent suit, for example. So our approach is really pragmatic and science-driven to classify HPAPIs from APIs and protect ourselves from these molecules.
So as said, Thibaud, in fact, we are a CRO and we are working for clients. So when we receive a molecule, sometimes we don't know, in fact, the OEL of this molecule. If we are lucky enough and we know this OEL, we can obviously calculate the OEB, which is the ranking, in fact, of the hazard potential of this molecule. You can have OEB1 for low hazard, till OEB3, OEB4, 5, and sometimes OEB6. So to achieve, in fact, this classification, when you don't have this OEL, you need to generate data. And as Thibaud says, we are lucky enough to have this toxicologist in-house. So by discussion in between the chemist and the toxicologist, we can define, depending on the therapeutical class, the pharmacology, the molecular analogy with something that is already known, the mode of action, and we have got also some in silico tools to guess, in fact, in which OEB will be this compound. But it's not all of that. In fact, we all have also to define or to discuss what will be the quantity that will be used. Is it gram scale, 100 gram scale, kilogram scale? Is there a dustiness potential? What is the direction of the task when you are going to use this powder? And with all this data in-house, we can find or adapt our strategy against the protection of this powder. Some example, in fact, of a compound that's got an OEL and an OEB that is known, nicotinamide, adapalene, prednisolone, or thalidomide. You've got something that is known in the literature. You can have directly the OEB, but what is the new API that we are going to manufacture? This is something that we don't know, and you will see in some example that in some case, like for example on retinoids, we can guess an OEB. We know for sure that this compound over there is a class 5 OEB because it's a very potent retinoid. And we know exactly, in fact, the structure-activity relationship of this molecule. We know which part of the molecule is responsible for activity. So we believe that the methyl ester here is also has to be taken as an OEB5. Anyway, if we go back one step below, on the boronic acid and the bromo-aryl derivative, we know that the structure is far away from the target compound, and this compound will be handled as a normal API. That means OEB3 in our strategy. On the other way around, if you're working with steroids, we know that this compound is OEB5 molecule, but as you can see, all the structure of the intermediate are really looking like the steroid molecule. So we believe that all the intermediate will have to be taken as OEB5 compound for the utilization of this molecule.
Okay, so now we see how we translate this into our different approaches and the various scales we are working with. So I will go very quickly on the gram scale handling of HPAPIs for class 4 and 5. I think there are a lot of commercially available devices and techniques that helps you to protect from these HPAPIs at this scale. One of the most important things to do, and this is true for any kind of scale actually, is doing first a risk assessment of all the critical steps you have to carry out with these molecules. So you can do that using this kind of matrices here. Another very important thing is the training of the people in the lab using these molecules and relying also on good internal procedures. But at gram scale and milligram scale, we perform all the route selection work, the synthesis of impurities, the solid phase studies, or the analytical method development. And for this, you can use simple system like closed vials for the reaction. We have crystal 16 devices where we can run solubility and crystallization screening in closed vials as well. We have this specific protective dome here that on our XRPD machine, for example, so we can run safely XRPD of highly potent molecules. And the handling of HPAPIs at this scale is commonly performed by a lot of labs in the world and by all our medicinal chemistry colleagues. So I want to emphasize more this second scale here, the 100 gram scale, the 100 to 500 gram scale, which is really the trick and the focus of this presentation. Because for this kind of scale, we have very few commercially available devices that are really adapted for the handling of these highly potent molecules. And it's also the stage where the manufacturing process of the molecule is still under development. So we need to go quickly to the delivery of the batches to perform the tox studies, but we also need to be flexible enough and agile enough to adapt to the variation we could give to the manufacturing process. So same thing as for gram scale. First of all, we do the risk assessment for all the operation. We took into account, as Jean-Guy explained before, the specificity of the early batches production, the limited number of operation, the limited batch size, the quantity that are manipulated, or the dustiness potential, for example. And for each operation, the weighing, the charging, the filtration, or the drying and the packaging, we did this risk assessment. And here is the solution we came up with. So this is a dedicated multi-gram scale highly potent facility. So it's a fully ventilated room with total exhaust, equipped HEPA filters, H14 filters.